Our next speaker is Professor Neeru Gupta, Head and the Stephen M. Drance Chair of the Department of Ophthalmology and Visual Sciences at the University of British Columbia and President of the International Council of Ophthalmology. Her presentation is titled The Eye's Lymphatic Clearance System and Implications for Disease. Please welcome Professor Neeru Gupta.
Thank you for the honor of being here, and thank you, Professor Lam, for the honor of the professorship and the invitation. It's a real privilege to be here among such esteemed colleagues, to be able to share some of the work that I personally have been very passionate about with my team. So the topic today is the eye's lymphatic system and implications for disease. But let me start with a reminder that 60% of our body is actually composed of fluid. And of that, 2/3 of that fluid lies outside of our cells. The other third lies inside our vascular system, very little in the CSF and the aqueous humor. So if we look at our circulatory systems, we all know the cardiovascular system, it's got a pump, the heart, it's a closed system, but we don't really talk about the lymphatic system. It's kind of an invisible system. It's a one-way system. It's open, and essentially it draws fluid from the extracellular space, and we've got a rich lymphatic circulation throughout our body, and it pumps several liters through our body every single day, at least 3. And this is what happens when the lymphatic system doesn't work well. So here you can see leg edema. It's a stagnation of the fluid, and very similar to when you get off of a plane after being there for a long time. So it's an important system. And it's important because it drains fluid, it removes waste, but it's also important for immune surveillance. So drawing on Professor McGee's talk, where he talked about passion and gaps and optimism, I'd like to take it from there. So we all know, based on our textbooks, that there are no lymphatics in the hair, there are no lymphatics in the nail, but it also tells us that there are no lymphatics in the eye. Well, if that's really true, how on earth do we keep a clear aqueous when we know that there's a metabolically active retina, there's a metabolically active ciliary body that spews proteins and waste into the aqueous every day, and yet the aqueous is clear? And so that was the challenge, and that was the spark, really, for the interest in this work that I really would love to share a little bit of a journey. And so the trabecular meshwork, we all know, is important for aqueous outflow. We all understand uveoscleral outflow is an important route. And by the end of this talk, I hope that you'll appreciate that we have an active lymphatic drainage system in the eye as a third outflow pathway. And so let me start with the first work. This was published in 2009, so it's over 15 years ago, where we identified what we coined as the uveolymphatic outflow pathway. And essentially, this is a diverse network of lymphatics that is located in the ciliary body of the eye. And I really wanted to share sort of those that sort of the eureka moment when we were looking at tissue in the laboratory. This is human ciliary body, H&E section, and this is the moment where we actually saw 3 distinct markers, one labeling the blood vessels, one the lymphatics, and the muscle. And this is the lymphatic vessel that can be clearly separated from the blood vessels, the electron microscopy, and the proof that these structures actually existed in the human ciliary body. Well, of course, we had to ask the next question, are they functional? Does anything happen to them? Is anything flowing through them? That's not easy, obviously, in a postmortem situation, and it's not, and these are very tiny structures, so we couldn't do it in the traditional models, and so we went to the sheep. Big model, the eye is about the size of the human eye, and we took radioactive tracer and nanospheres and injected it into the eye, and 15 minutes later sacrificed. And what we found was we found nanospheres inside labeled lymphatic structures inside the ciliary body. And so this was all part of this work. And for those of you who submit papers that get rejected again and again, don't give up. This too was rejected. Grants were rejected. So carry on. The future is bright. Stay optimistic. Stay with it. So this was the first paper that really led the way and paved the way for some of our subsequent work. We were very interested in measuring lymphatics drainage from the eye, and we referred to an early work by Jerem in which patients on the ward were their lymphatic ducts were cannulated, jugs were placed by gravity drip to figure out what was going on inside of these, what was the composition of the lymphocytes or leukemic patients. And so we used the same approach, and we did that in sheep where we put them on their back and we cannulated them, and we were able to use this as a model to measure lymphatic drainage from the eye, and so we know that it is possible. And so there is a rich uveal lymphatic pathway outside, but what do we know about the pharmacology? Well, lymphatics in the body, for example, in the mesentery, have a rich pharmacology, and we know, for example, that PGF2 alpha stimulates the lymphatics in the gut. So we thought, well, PGF2 alpha, that sounds pretty familiar. We've got one of the most common drugs for glaucoma that we use to treat it. So why don't we have a look? So we turned to the mouse model. And in this model, what we did was we used quantum dots. We injected quantum dots into the eye with the understanding that if they do flow into the lymphatics, we should be able to collect those quantum dots and see them in lymph nodes. And we used a technique called hyperspectral imaging. The benefit of this is that the mice stay alive. And so this is pre-injection of the quantum dots. Here, after the injection, you can see a red spot in the eye. And this is what happens if you just leave it alone and sequentially image this live mouse. 6 hours later, you see a red spot in the neck. Well, we know that this was a lymph node. We did the histology, and you can see the quantum dots sitting inside a lymph node. So we know that if you just take a mouse, you inject quantum dots into the eye, you wait 6 hours, boom, you'll see it in the submandibular node. But what happens if you pre-treat with PGF2 alpha or prostaglandin analog? This is what happens 20 minutes later. So within 20 minutes, we move from the eye to the neck. So we were able to show that latanoprost actually stimulates ocular lymphatic drainage from the eye. So we were pretty excited about sort of this early look at the pharmacology of the eye. And then, of course, one of the questions is, can we measure lymphatic drainage in response to drugs? And so for this one, we used a different approach called photoacoustic imaging, where we used pulsed laser light, we used ultrasound, and again, this technology allows us to keep the animals alive so that it's in vivo throughout. And we created a dye that could be picked up and used this to measure the speed at which tracer left the eye and the speed at which it enters the submandibular lymph node, and through that correspondence measure quantitatively what was going on with some near-infrared imaging. And what we found was that there was active lymphatic drainage from the eye that could be measured by non-invasive photoacoustic imaging of near-infrared nanoparticles. So essentially, we had a model, we had an assay to measure what goes on. So we applied this assay to to study what was happening in aging over time, and what we found is similar to diseases like glaucoma. There's a age-related decline in lymphatic outflow from the eye. We were curious to see what happens with beta blockers, so we used this assay to show and found that beta adrenergic drugs actually reduce lymphatic outflow from the eye. And so there have been a slew of works over the past decade to help us understand, both identify, characterize, quantify, and develop assays to understand the system, which we hope will be targetable for purposes of treating eye diseases that are common to many. One of the things that we were particularly excited about is the lymphatic system. So this work led us to question the way that CSF fluid drains. And what we found is that the optic nerve actually has a rich lymphatic system. And this is a slide that shows a sleeve of CSF that's entering through a blood vessel inside the parenchyma of the optic nerve. And this is the first description of a lymphatic pathway in the optic nerve. This is a very important system in the brain to remove waste and detoxify. It's considered important in Alzheimer's disease. And we have also shown that this is reduced in a glaucoma model. And so it raises all kinds of questions. What is happening when the system is not working well, right? So while we understand that the brain is important and that the eye is only a fraction of what we see in terms of, let's say, for example, take a retinal ganglion cell journey. Most of it actually lies outside of the eye. And the fact that we've got CSF and the brain interacting and how important it is for the eye. And so these open up all kinds of questions about what should we be understanding about that might help us understand things. From a tumor point of view, we all understand the spread of tumor. Is there an opportunity for us to understand how this spreads via lymphatics through future studies? Our most recent interest lies in SANS, which is a very important disease that prevents us from long spaceflight mission. And so this is a post-flight mission, and you can see that intraocular pressure and retinal nerve fiber layers are changing. In summary, there's a lot of implications of the system. We hope that they'll be targeted, and this will be the groundwork for future studies. Reminder, next time we look at the eye, consider that there's fluid. There's a lot that we can't see. And with that, I'd like to acknowledge my colleagues and collaborators and all of the funding sources. Thank you very much for your attention.
Thank you.